Hello students, we have learned about the hybrid course, laws of inheritance extracted from the hybrid course and chromosomal theory of inheritance. Now we are going to learn about linkage and recombination, and also we are going to learn about sex determination and mutation. You know, Thomas and Morgan carried out several dihybrid courses in fruit flies to study genes that were sex-linked, means the genes which are present in sex chromosome. I think you already knew that we have two types of the chromosomes: allosomes or sex chromosomes and autosomes or other chromosomes. You know, one pair of chromosomes, that is the pair of X and Y chromosomes, is sex chromosome or allosome, and other 22 pairs are autosomes or other chromosomes. The genetical courses done by Thomas and Morgan and his group were similar to the dihybrid courses carried out by Mendel in pea plants. For example, Thomas and Morgan and his group hybridized yellow body, white eye females of Drosophila melanogaster or fruit flies to brown body, red eye males, and intercourse their F1 progeny. He observed that the two genes did not segregate independently of each other, and the F2 ratio deviated very significantly from the 9:3:3:1 ratio. Morgan and his group knew that the genes were located on the X chromosome and saw quickly that when the two genes in a dihybrid course were situated on the same chromosome, the proportion of parental gene combinations were higher than the non-parental type. Morgan attributed this due to the linkage or physical association of the genes and coined the term linkage. The definition of linkage is the physical association of two genes in homologous chromosomes is called linkage. You know, homologous chromosomes means the identical chromosomes. Suppose we human beings have 23 pairs of the chromosomes in each cell. Out of these 23 pairs of chromosomes, 22 pairs are identical and homologous, and one pair is not identical. X and the Y chromosomes are not identical, but if it is in case of the female, all the 23 pairs are homologous. In these pairs, half the number of the chromosomes are from father and other half is from mother. I hope you have already understood that genes are located at the chromosomes, and the regions of the chromosomes where genes are present are called loci. In singular, we call it locus, right? You also must have known that there is crossing over of chromosomes during gamete formation by meiosis. In crossing over, there is exchanging of genes in the homologous chromosomes. Genes from father's chromosomes and that of mother's chromosome take exchanging during gamete formation or gametogenesis. The process is called recombination. By doing so, every newborn babies become variated in characters from their parents, means they do not become same copies of father or mother. If there is no recombination in some parts of the homologous chromosomes during gametogenesis, then after fertilization, the embryo may become same copy of its parent. This phenomenon is called linkage, right? You know, two genes that are located close to each other on a chromosome are often inherited together, in which the process is called linkage. In contrast, genes located farther away from each other on same chromosome are more likely to be separated during recombination, the process that recombines DNA during meiosis. Okay, let us have a look at cross, which was done by Morgan, T.S. Morgan and his group. Okay, two crosses are given here, cross A and cross B. These crosses were done by Morgan and his group. Okay, let's look closer to this cross A. This is male Drosophila which have wild type trait, and this Drosophila is a female Drosophila which has yellow-white. Wild type is nothing but it is a brown body, red eye. Y plus W plus genes are dominant over Y W genes. This is X chromosome. This is X chromosome. This hook-like structure is nothing but it is a Y chromosome. Y chromosome is not homologous chromosome of X chromosome. Means Y chromosome is not identical with X chromosome. So in F1 generation, we Morgan and his group got wild type and yellow-white offsprings. Yellow-white offspring is male. This Y chromosome is from male parent. This is X chromosome. It is from female parent here, YW. This X chromosome which carries Y plus W plus genes is from father. This YW genes are from mother. So two types of individuals are produced in F1 generation. When these individuals are fertilized, let's see what happens. If there is no crossing over, this X chromosome which carries YW genes and this X chromosome which carries Y plus W plus genes, these two chromosomes are not in crossing over, then 50% of the female gamete will carry Y plus W plus genes, and other 50% of female gametes will carry YW genes. So let's see the offsprings without crossing over. Four types of offsprings are here: wild type, yellow-white, wild type, yellow-white. This is hook Y chromosome is from father. This Y plus W plus genes are from mother. So it become wild type. This Y chromosome is from father. This X chromosome is from mother, YW. So here, this X chromosome which carries YW is from father. This chromosome which carries Y plus W plus is from mother. One is from father, one is from mother. So these are parental type. Parental type means by linkage. Chromosomes are not segregated. Y plus W plus genes are not independently segregated with YW genes, right? If there is crossing over, here, Y plus will be assorted with W. W plus gene will be assorted with Y. So after crossing over, we got white. Actually, white is nothing but brown body, white eye, yellow. This Y chromosome is from father. This Y plus W is from mother. This female. After crossing over, Y plus W. Crossing over has taken already. So this Y chromosome is from father. This
X chromosome is from mother after crossing over. This X chromosome which carries YW genes is from father. This actually in male there is no crossing over. Y chromosome and X chromosome is not having cross. Y chromosome and X chromosome are not crossing over because they are not identical chromosomes. Y chromosome and X chromosomes are not identical chromosomes. They are not homologous. Okay, this X chromosome is from father and this is from mother, F1 female. This X chromosome which carries Y, W plus is from F1 female. This is from F1 male. So these are different non-parental type: white, yellow, white, yellow. So recombinant is recombinant types are only 1.3%. Parental type are 98.7%. This is linkage. Okay, students, for cross B, you can study by yourself.
Okay, let us see what is the recombination. Recombination is a process that produces new combination of genes by interchanging of the sister chromatids of homologous chromosome. Okay, students, Thomas and Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were very tightly linked, while others were loosely linked. Okay, now sex determination. Establishment of sex through differential development of an individual at an early stage of life is called sex determination. Sex determination is of 4 types. Sex determination by environmental factors, non-allosomic determination of sex, allosomic genetic determination of sex, and haploid diplontic sex determination. According to your syllabus, we will learn only 2 types of sex determination. Allosomic determination of sex, simply we call it chromosomal sex determination. Another one is haploid diplontic sex determination. So firstly, let us learn chromosomal sex determination or allosomic determination of sex. In 1891, Hermann Hanking discovered a nuclear region which was known as X body. Now we call it as X chromosome. The term X body was given by McClung or MC Clung. When Hermann Hanking carried out experiments on fireflies, he discovered the X body. Further investigation by other scientists, Steven and Wilson, led to the conclusion that the X body of Hanking was in fact a chromosome, and they named the X body as X chromosome. So chromosomal sex determination is of again 3 types: XO or X0 or XXX0 type of sex determination, XY type of sex determination, and ZW type of sex determination. Firstly, let us learn what is the XO type or X0 type sex determination. In this type of sex determination, the female has a pair of X chromosomes, a pair of homologous X chromosomes with autosomes, and the male has a single X chromosome with autosomes. Male has a single X chromosome with autosomes. This type of sex determination is found in a large number of insects like grasshopper, fireflies. In this type of sex determination, all the ova or female gametes or eggs bear an additional X chromosome, while 50% of the sperms bear X chromosomes. The eggs or female gametes fertilized by sperms having X chromosome become female, and those fertilized by sperms that do not have an X chromosome become males. Okay, second type of sex determination is XXXY type, or simply XY type of sex determination. This type of sex determination is found in Drosophila melanogaster and mammals including human being. Male individual is a heterogamety, that is, sperms have different types of allosomes or sex chromosomes. 50% of the sperms carry X chromosome, other 50% of the sperms carry Y chromosome. If the fertilization occurs between the egg cell with the sperms carrying X chromosome, the individual will become female. If the fertilization is between the egg cell with the sperm cell carrying Y chromosome, the individual will become male. Half of the sperm cells carry X chromosome, other half of the sperm cells carry Y chromosome. That is why the condition of XY type of sex determination is called male heterogamety and female homogamety, because 100% of the egg cell or female gamete carry X chromosome. Okay, third type of chromosomal sex determination is known as simply ZW type or ZZZW type of sex determination. This type of sex determination is found in some birds. This type of sex determination is female heterogamety, male homogamety, because female gametes produced are of two types. Half of the female gametes will carry Z chromosome, other half of the female gametes will carry W chromosome. Okay, now we have haplodiploidic sex determination. You have already understood that honeybee is having one special type of reproduction, that is parthenogenesis. Parthenogenesis is a special type of reproduction in which the female gamete directly develops into new individuals without fertilization. You know, gametes are always haploid. If the new individuals are developed directly from the female gamete without fertilization, without fusion, then new individual will be haploid, right? Because there is no fusion, there is no formation of diploid, right? So in the process of parthenogenesis, honeybees develop as haploid organisms. If there is a parthenogenesis, honeybees are always male. If I ask you what is the ploidy of male honeybee or drone, then your answer will be haploid, because male honeybees are always developed from the female gamete without fertilization in the process of parthenogenesis. If there is fertilization, the new individuals will be diploid, because there is a fusion of two haploid cells, one sperm cell, one egg cell. If fertilization occurs in honeybees, honeybees become female. So the ploidy of female honeybees, worker honeybees or queen honeybee, is diploid. So female honeybees are always diploid, male honeybees are always haploid. So the type of sex determination in honeybees is called haplodiploidic determination of the sex, right? Okay, now let us learn what is the mutation. So what is mutation? Mutation is a phenomenon which results or causes alteration of DNA sequences, resulting in changes in the genotype and phenotype of an organism. There are different types of mutations: gene mutation and chromosomal mutation. The gene mutation is of two types: point mutation, Frameshift mutation. What is a point mutation? It is the type of mutation which involves a single nitrogen base change, One example is sickle cell anemia. About sickle cell anemia we will study later in this chapter. Now other type of mutation is called Frameshift mutation. The type of the mutation which arises due to change in some base pairs is called Frameshift mutation. You know, Frameshift mutation is due to delation, duplication and insertion of base pairs of DNA. Chromosomal mutation is of two types: structural mutation and a numerical mutation. If the chromosomal mutation is due to change in the structure, it is called structural and chromosomal mutation. Structural and chromosomal mutation is caused by delation, insertion and duplication of some parts of the chromosomes. Next type of chromosomal mutation is numerical chromosomal mutation. Numerical chromosomal disorder or a numerical chromosomal mutation is of two types, aneuploidy and polyploidy. Aneuploidy is the type of numerical chromosomal mutation in which there is gain or loss of one chromosome in the genome of an organism. It is again of two types, monosomy and trisomy. If there is a loss of one chromosome, that type of aneuploidy is called monosomy. If there is a gain of one chromosome, that type of aneuploidy is called trisomy. Now polyploidy. It is a chromosomal mutation in which there is the addition of one set or more sets of chromosomes in the genome of the individual. It doesn't occur in animals, it occurs only in plants. The factors which cause mutations are called mutagens. Mutagens may be environmental factors, example UV ray, X-rays. These are the environmental factors which cause mutation. Biological factors are also there. One example is the retrovirus. Okay, we also have chemical factors which cause mutation. Some examples are acridine, mustard gas, then phenol. Okay, students, let's do this much for today. I hope you have understood about linkage and the recombination and also about sex determination and mutation.